Okay, good afternoon, everyone. So, just picking up what we have left the other day on Monday. So, I said actually there are lots of examples, even in these days, working as a ribozyme. So, the RNAs, but the usual, our stereotypical RNA's function is simply playing a second fiddle of taking DNA information as a genetic information and then deliver. Okay, this is what the DNA said, so do as what it said, okay, or do as what she said. Simple. But actually, it turns out that so many important enzymatic functions are carried out by not protein because that's another stereotype because a vast majority of enzymes are actually made of proteins. There's a reason why proteins can only function as enzyme. It's a shape. Protein has a chemical property that can fit into and adapt into so many different unique shapes. Shape matters. Shape dictates function. There's no more detailed or complicated secret or magic involved in trying to figure out the function, any biological function. One particular function is done because that's what it's supposed to. So because of the shape during the chemical reaction, for example. Yeah. So anyway, so being able to generate unique shapes are extremely important, critical. And unfortunately, proteins are the only ones that can generate so many different shapes. Like proteins, freshly out of newly synthesized protein from this guy, the ribosome. Ribosome is a machinery from which proteins are synthesized. And this protein can fold whatever they want. You can fold this protein like this shape. Be careful, I may lose my iPad and then drop it. And this kind of long, slender shape. Or in some occasions, depending on who you are, some other protein have this ball-like shape. We call it the globular proteins. So many. It all depends on the DNA sequence actually, because there is a strict and direct specific relationship between DNA as a gene and a protein. The kind of mechanism by which DNA serves as a genetic material is it dictates or it determines what kind of proteins are going to be made from a certain area of the DNA. Then if RNA simply do a good job of copying that information and deliver to this guy, the ribosome, and this ribosome is a big guy, no, could be a lady. A big ribosome can interpret the recipe. Okay. What did I say about this whole thing, whole business? I compared the cooking. DNA is simply providing a recipe for specific dishes. RNA is the one particular dishes. DNA is probably a whole cookbook, entire cookbook containing probably 25 to 30,000 of different recipes. How to make each different things. And then each different RNA copy as in the form of a convenient form of this recipe, okay, instruction, then delivered to hand that recipe over to this big structure called the ribosome. And the ribosome can read that instruction. And then ribosome can actually turn. So up until then, there was everything was simply just the informatics. There's no tangible things has yet involved. Now this ribosome can turn those information only thing into real tangible thing like proteins. And now you get to produce so many different proteins. And proteins are, by the way, made of or amino acids, different amino acids. And the beauty of proteins in that they can generate so many different shape, simply depending on the order, amino acid order, because there are so many different amino acids, chemically speaking, and there are 20 such amino acids that participate in. So you imagine you have 20 different amino acids to choose from. And let's say, for example, mathematically speaking, if you had to assemble a protein, a very simple protein, in fact, having only 100 amino acids, which is a very small, simple protein. And then how many different possibilities you can build if you place each amino acid in an orderly fashion out of 20 different things. There are so many different ways you generate, even with only 100 amino acids. Let's say if you were to make a protein consisting of 1,000 amino acids, there are much more possibilities that you can build so many different types of a protein. So in this way, you can generate so many different shapes of proteins, and each protein so that function uniquely in each specific different ways, including enzymes and antibodies and receptor proteins, transporter proteins, so many different things. Okay. That it all depends different shape. Now, the RNA can mimic such thing because RNA, for some reason, which I don't want to go into too much detail, but anyway, believe me, trust me, RNA can also generate such different unique shape, unlike DNA. DNA is such a boring guy. Yeah, I can call it DNA a guy because DNA is so boring. So DNA is such a boring structure, but unlike DNA, although chemically DNA and RNA, if you try to think about it, they are so similar. But for some reason, RNA, just subtle difference present in RNA allow RNA can behave as to be able to form a unique shape. So that is a kind of a prerequisite for RNA also to be able to function as a kind of enzyme. And then it showed that simply, yeah, there was actually presence of such a RNA-based enzyme exist inside of our life. And that's called ribozyme. And I just preached the importance of this guy called the ribosome. Okay? This ribosome, the big structure within which proteins are made, and actually the active ingredient. This is a whole complex mixture, but the most important function of this ribosome is to be able to assemble different amino acids so that generate actually final product of a protein. By the way, why do I have so little audience today? 1, 2, 3, 4, 5, 6, 7, only something like 20 students out of 74 students. Wow, great. Today is what? Do we have some kind of festival? Oh, yeah, that explains. Good. So, where was I? This ribosomes, the most critical function is to assemble each amino acid to generate the final product, which is protein. But that function, it should be catalyzed by enzyme. So you may think that because this whole mixture within which so many proteins are actually inside of this ribosome, but not a single protein can do this job. But actually, it turned out that surprise, there was RNA. RNA was the one who actually was responsible for assembling, putting those amino acids together. Okay, so the RNA's importance. This is one of the most obvious, striking showcase of the ribosome's importance. Anyway, then why not simply, RNA was such a multifunctional, there was a good guy, RNA. It looks like RNA is much more capable than DNA. Then why didn't we switch from RNA into DNA as a genetic material? That was another question. Because you said in the beginning, there was an RNA acting as a genetic material, but DNA was nothing. But then eventually DNA took the part and RNA was pushed away. Okay, so from now on, I'm in control. I'm in charge, and DNA became the genetic material. Why? Because this stability is it. Although RNAs are so versatile, but chemically, if you let RNA sit on this room temperature bench for 20 minutes, and then it will spontaneously disassemble itself, RNA whole thing, into pieces. On the other hand, DNA is chemically so stable. No wonder why and how, even if you found some fossils remaining of the bones of Neanderthals, and then still from out of it you could extract DNA, and then you restore whole thing, and then they decoded the entire genome of Neanderthals in these days. DNA is that much stable. It can be stored for a long time. It has a high resistance against so many different chemical attacks. Unlike RNA, RNA is so unstable. So if you are trying to consider one suitable chemicals as your genetic material, probably stability is probably the top priority you should consider. How can you trust your whole thing, everything, your essence, on anything which has such a vulnerability for unstable thing? So the minute you could escape from RNA, then you just simply grab the opportunity and then you just ran away from RNA and then stick with the new guy, DNA. Probably that's what must have happened. So although RNA was so helpful, without RNA in the beginning, the life would not have been possible, because that was the only one who could just juggling things between the genetic material and the enzymatic function. Okay, but once your job is done for particular period, okay, then with so much thanks, then you should go away and then probably find another job, which nowadays you find. RNA in these days, of course, is such an important material, chemicals. RNA is doing so many different important things in maintaining your genetic integrity. So I'm not saying RNA is unimportant in these days, but simply there was a speaking only of the DNA, I mean the genetic material, who is the better, more suitable to become a genetic material? Obviously, DNA. So the reason why is that RNA, because of there are subtle difference. One of the subtle difference is the presence of oxygen in RNA in this particular position. DNA doesn't have an oxygen in this position. And the presence of that oxygen is such a headache, because oxygen is always a villain. Always, whenever oxygen is there, then the oxygen is trying to do something. Oxygen is so reactive. Try to take any electrons away from the other chemical. So it will attack, and click, and break this chemical bonds. And then even considering within this very small area of RNA, long strand, RNA became spontaneously come into pieces spontaneously. DNA doesn't do that because DNA does not have the oxygen on that position. So DNA is good. I mean, stable. Okay, so different types of RNA. Later on, probably we are going to take a look at, what they do. What do they do individually? These three RNAs are the conventional types, three types of RNA. Probably you had learned in your middle school or anything. But there are more, more different kinds of RNA exist. Okay? They are all relatively recently discovered. So I'm not going to even mention those other types of RNAs, but there are many, many different, not many, 3 or 4 more different kinds of RNA do also exist, involved in many important cellular functions. Okay. Having said that, try to jump into now the actual evolutionary things. So up until now, I have explained the chemical evolution, right? Now, once you have succeeded in chemical evolution, now everything else from then on is through the biological evolution. The biogenesis, through which this particular regulatory mechanism we recognize as biological evolution or Darwinian evolution. Because the reason why someone actually asked why this biogenesis is called the Darwinian evolution, as opposed to, try to make it in contrast to the chemical evolution, because through biogenesis now you generate diversity, okay? And each diversity is a subject to the natural selection. Okay? So that's the kind of a key principle concept that Charles Darwin was looking at. So that's why it's fair to call that as a Darwinian evolution. And so I have repeated this procedure so many times until now, so I won't have to repeat this thing again. You can just simply go over later on when you have time to sit down and review for your upcoming exam. Okay. So here, the variation, as I mentioned in the last lecture, the ultimate source of this whole variation, different variation, of course, there are many different tricks you can facilitate, increase the diversity. The sexual reproduction is one such a way. Through sexual reproduction, as opposed to asexual, simple keep dividing, you can tremendously increase the diversity. There are some specific reasons why. Okay, but anyway, no matter what you do, that's all just a trick. The ultimate source of all variation you have to generate at the DNA level. If you can understand the DNA as a whole bunch of the DNA sequence, how many of you have heard of something called a DNA sequence? Raise your hand. How many of you not heard of it? You have never heard of something like a DNA sequence? Raise your hand. Thank you. At least you are honest. Okay. So everybody, at least everybody has heard at least once or what something called DNA sequence. Although not many of you exactly understand the true meaning of DNA sequence, though. Okay, it's okay. But so DNA is simply the way DNA function as like genetic material is the key secret reside on so-called DNA sequence. It's just like alphabet. Let's say you are trying to read a book written in totally different language. You have never been exposed to that language. But surprisingly, that language, the whole language, contains only 4 alphabets. In English, there's something. How many alphabets are there? Are 26 alphabets or anything in English? Okay. In Korea, there are 24 alphabets. So at least some, aside from Chinese, that is totally different, the systematically. But although these different languages have some certain number of alphabets, different alphabets. So DNA acting as a genetic information. Gene is simply an information. Yeah, although I jokingly compare this as some kind of recipe, but if you think about it, it is actually acting as some kind of different recipe or instruction. Yeah, this is how you make certain particular protein. So it is simply just information. How are you going to present the different information, like different recipes of in human? There are approximately 25,000 different such recipes exist. Okay. So you can generate 25 different foods, dishes you can generate. Actually, in reality, you can generate 100,000 different dishes out of 25 recipes. That is another trick, but yeah. Anyway, so those information juggling with only 4 different alphabets is amazing, right? And then trying to make sense with those, simply keeping and having different combination out of those 4 such alphabets. So mutation is, so you in a certain region, let's say this is a long strand of DNA where there are certain specific narrow area which is specified. Okay, let's say this is the only area specifying a specific gene, which means this area will signify or designate a specific protein, maybe hemoglobin, HB. Hemoglobin protein is made from the instruction written within this area. That's one such concept. And this information is written by a whole combination, all different random looking combination of 4 different letters of DNA. You may wonder how, using only 4 alphabets, can you generate some really meaningful and complex information? There is a way, don't you worry. Yeah. So there is a way doing so, but the important thing that I'm going to point out today is, so there must be some specific orders of a combination of these 4 alphabets is extremely important to make sense and then stay intact. So if you simply disrupt that fine order of those alphabets, then you are in trouble. This whole area originally was written for, hey, here is how do you make this hemoglobin protein. But if you somehow change some order, specific order, then that guy, the ribosome guy, wrongly interpret, it says, I thought this instruction bring some particular amino acid, but it says they have changed their mind, bring another amino acid over here, and then it will. Yeah, the chances are it can be generated. A totally different type of protein can be generated. That's what we call the mutation. So mutation always begins with such change in DNA sequence. So once again, then we can define correctly what's mutation. The mutations are changes in the DNA sequence. Yeah. If I simply abruptly throw this definition, probably some of you are list. What? What's DNA sequence? What's got to do with this functional thing? Maybe. Yeah. That's their relationship. Sequence. The DNA sequence, what we call is simply the order of such a DNA alphabet. By the way, DNA alphabet, A, T, G, C, 4 different types of alphabets are there. These are the ingredients, raw material from which whole thread like long DNA is synthesized. So it will be simply repeats of different combination of this whole 4 different types of DNA alphabet is your DNA sequence. So, and last lecture I also said every time you try to replicate your DNA, there's no way you can avoid this mutation occurring. Yeah, it will. Mistakes happens always, right? Just like when you try to dictate the long sentence on your computer, you always make typographical error, and the enzymes replicating DNA will do the exactly same. Yeah, they are also human. No, they are not human, but they are the component of human, so they will make mistakes. There is no such thing like a mistake-free. So those mistakes are the sources of mutation. Here, at this point, there's no point of going over this art chemicals. So totally disregard, ignore this. Only a few of you sitting here can understand what this means. So forget it. So such alteration, of course, can be accelerated by many chemicals. Those are called carcinogens. Why carcinogens? Carcinogens, most of you have thought carcinogens are the ones chemicals that causing what? Cancer. Yeah. But actually, the real definition is carcinogens are, of course, if you ask this thing into doctor, of course, medical doctors will always say, hey, carcinogens are those chemicals causing cancer. If you ask this thing to any molecular biologist, then carcinogens are any chemicals that increase this rate of mutation. That's the relationship between, yeah. If you increase the mutations, then the chances that you could also change the very critical genes DNA sequence. Those critical genes happen to regulate your normal, so to speak, cell cycle. What is this cell cycle? It's not bicycle. The cell cycle is a regularity of cell division. So if you disrupt those important genes involved in this cell cycle regulation, then these cell cycles go out of control. So it will keep dividing. Now, out of control, keep dividing cells are what? Cancers. So that's, yes?
What difference of carcinogen, mutagen, and teratogen?
Various. Two molecular biologists thought there are no differences. Teratogens are actually some more specifically you can define as those chemicals that cause mutations so that you have some deformed bodies like babies. And so those are specific teratogens. But many such teratogens are also carcinogens, of course. And all this, the mutagens. So maybe differences are there actually. Good questions. Let's say this big circles are mutagens, chemicals. Then small portion, exactly carcinogens. Okay. And some of them maybe overlap or not, so teratogen. Okay. So those are probably some of the distinction. So the mutagens are the largest circle, within which some of them are more specifically having some effect in cancer causing, that we call them carcinogens. But usually, so most of those chemicals that increase the mutation rate, we usually have a suspicion, strong suspicions that that could be also carcinogens too, to the view of a molecular biologist at least. Not to the doctors, of course. If you just ask this thing to the medical doctors, they will, hell no, what are you talking about? That's what probably he would, she say. Okay, so 6 common carcinogens, no, I don't have to go over. So this, ignore this particular one, you don't have to worry about. So even if, let's say, here is the logic. Even if you had single ancestor as a Luca in the beginning, that was the up until you achieved the chemical, succeed in chemical evolution. So through the chemical, unlikely chemical evolution, let's assume that you were able to somehow manage to generate your first type of a cell or as an ancestor. From then on now, this cell, true cell, should be able to divide and reproduce, okay? It doesn't matter, I don't care whether you do this through asexual way or sexual way, it's just, it's up to you. Now, you keep dividing. So it produced so many progeny now. Then through this mutation, right? That mutation inevitably will soon after generate so many diversity. So once you generate diversity, some of those diverse type having different traits will have some different preferences. So, well, okay, from now on, I'm going to go with this sexual reproduction. It seems like more attractive to me. Some other type of the type of organism, well, I don't like that kind of a hassle. It's too much. I will simply keep my simple life. I will keep dividing, and then more efficiently, economically, reproduce. That's your choice. Okay. So many diversities are generated and keep going, and being subjected to equally to a different selection force by the natural selection, right? So here we are. But in the meantime, during which some of the particular different type of forms or mode of lifestyles, okay, that I'm going to talk about. Once you have generated the first lines of true life forms as a cells, okay, are there some different types of cells? Of course, prokaryotes and eukaryotes are such, of course, different types, of course, obviously. There are big time lapse between, although the first prokaryote generated versus the first eukaryote generated got separated from more than 1 billion years. It was a huge gap. But anyway, speaking of such different types, first let's try to recognize this mode of nutrition. First, heterotrophs. Because the type of organism depends their nutrition acquisition on this. We call heterotroph. Hetero means different. Trop means roughly you can translate trop as a food. So the correct definition of a heterotroph is actually this. Those who have to obtain their food only through other organic materials. Organic material. In other words, heterotrophs always have to obtain their food in the form of organic material. Then you may ask, Are there any other forms that actually they can obtain their food in non-organic, which means inorganic material? Yes, there are. There are. It's amazing though. So we are heterotrophs, by the way. So the food we use, the food we consume, or the material we consume as foods are all organic material, if you think about it. What did you have for lunch today? Oh, you skipped lunch for diet? Good. What did you have for your lunch? If you think about it, anything you ate for your meal must have been organic. Of course, some drink containing basically inorganic things, but that's just a drink, a refreshment. Food, by definition, is the one who can or which can give you energy. That's why you need organic material for your food. The usage is once you get this food material inside of your body, you have to break down, you have to break those through the catabolic reaction. Then what do you get? Energy is released through catabolic reaction. That's the purpose. You will get the energy, capture that energy, and then use those energy in many other area that you need. That's the definition of food. So food must be organic, obviously, of course. But surprisingly, there are. So here are some big examples of heterotrophs. The one thing is, in the beginning, all those life forms as the first ancestral form of the life, all of them were heterotrophs. Okay. So let's say the first life form was bacteria like prokaryotes. They were all in the beginning all heterotrophs. And animals and fungi, nowadays, of course, they came much later throughout the evolution, of course, but they are heterotrophs. And this protist, okay, also another eukaryotes, okay, more simple life forms of eukaryotes, most of them are also heterotrophs. Then what are the other ones? The opposite. Between these animals and fungi, although these two are heterotrophs, but there are subtle differences in that animals take their food first, and inside they take care of this digestion. But fungi, they digest their food first outside and then absorb it. That means fungi don't have any the organ like a stomach or digestive organs. They don't need because they already digestion took place outside of their body. So, but that's not an important distinction. So anyway, one bottom line is, as a heterotrophs, the sad reality and the sad fate of any heterotrophs, where can you get the organic material? The source of organic material. If you search for soil, can you get organic material? No. The organic means organism. So the only place you can find the organic material is the body of other living. Those who still living or those who at least who once lived, now dead. That's just really shitty, I'm sorry, the sad fate of all heterotrophs. You have to take other lives in order for you to survive. That's heterotroph. But you don't really have to do that. If you can become autotroph. Autotrophs, the only difference between hetero and auto is now their food can start from inorganic materials. That's the big difference. They have an ability of converting this inorganic material in their body into the organic material. When you try to imagine autotroph, you just immediately, probably a lot of you immediately, it occur to you that, oh, those photosynthesis. Of course, photosynthesis, those who can do this photosynthesis as a metabolism are autotrophs. What's photosynthesis in Korean? Everybody knows, right? Okay. That's an autotroph, yeah, but photosynthesis. Yep, okay. So you guys don't know? Oh, shame on you. Yes.
The plant that like Dodder, Lenoa, and Indian pipe plant that 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 can't photo can't auto draw.
What? I'm sorry.
풀 가운데
Uh-huh.
이거 야구라든지
Uh-huh.
Dodder, Lenoa, Indian pipe plant처럼
Uh-huh.
스스로 합성하지 못하고
Uh-huh.
생산한
The parasitic plants are there, yes.
이건 헤테로트로프인가요? 어떻게
Yeah. Well, if they lose entirely of their ability of this photosynthesis and then simply feed on as a parasitic plants, then now they technically should be considered as a heterotrophs. There are lots of plants, yeah, living on other plants, sucking up other plants' photosynthetic thing. But other some semi-parasitic plants, they still have their ability to do this photosynthesis, but then still they have their own choice. No, I don't want to do this. I will just rather suck it from others. Just facultative parasitic plants are also there. It's too much. Okay, ignore this. So anyway, the big difference between auto and heterotroph is that these autotrophs can convert. So here, that is the reason why CO2 plus water, voila, simply C6H12O. This is a chemical formula of glucose, okay, by the way. So this simple inorganic materials are being converted into organic material, organic compound, which is glucose. So what is this? Is this anabolic or catabolic reaction? Making glucose starting from carbon dioxide and water. Anabolic? Yes, anabolic, right. Because how do you distinguish anabolic from catabolic? It's very easy. Whenever you see some more complexity, even if that complexity is involved with only one simple element. So if you start from the simpler, smaller material to become larger, more complex, then it's always anabolic. Going opposite is always catabolic. And the energy direction, whenever you try to more complex, larger, it takes energy. Because that's a non-spontaneous reaction. That's anabolic reaction. That's how you distinguish these two. So that means, obviously, if you want to do this inside of your body, you have to find a way to get the energy required for this. That's why they pray for Almighty Sun. Okay, give us energy. So that's how they learn to this photosynthesis. Okay. So photosynthetic organism is not the only autotroph, of course. There are many different several other different way of doing this autotrophs, but to our eyes on the surface in these days, it looks simply photosynthesis is the only example that we can find as an autotrophic metabolism. But if you really dig into some small area, like deep in the sea or deep in the in the earth, you will be able to find so many bizarre other different way of metabolic things. So anyway, Now, the second point is this. So having said that, so starting from the first common ancestral form of life, which I said all of them are the prokaryotes, and all those prokaryotes were what? Heterotrophs. Okay? So the origin, about 4.6 billion years ago, the Earth began forming its present shape. And about 1 billion years later, the first, not fist, shoot, fist, the first, sorry. The first prokaryotic organisms appeared. How do you know? How do you know that actually around 3.5 billion years ago there was the first ever form of prokaryote life appeared on Earth? Obviously, you were not there. Then how do you know? So the only clue that you can say this is fossil record. So you somehow luckily was able to find a fossil of bacteria. You've been extremely lucky. Wow. And then it turns out that so you just figured out how old is this. And then you just estimated that, scientifically estimated. It turns out, oh, shoot, it's about 3.5 billion years old. So if you found later on another fossil that is older than, another fossil of prokaryotes older than this 3.5, let's say like a 4 billion years old fossil of a prokaryote bacteria cell, then you have to change the theory, present theory, into the new one. But currently, yeah. So then let's try to compare. The first eukaryotic organism in the form of a fossil that you have discovered is only 1.8 billion years ago, so there is a huge gap. Labs exist between the prokaryote and eukaryote. That means probably your best guess, your best educated guess to explain this probably would have been, okay, so eukaryotes are all must have evolved from at least some small branches of prokaryotes. But the important things today that I'd like to emphasize is, because today, obviously, I'm not going to be able to finish all this in this short today's lecture. Well, lecture time is always too short. Is oxygen is accumulated now, during which. That means in the beginning, there was no oxygen in the Earth's atmosphere, in the gaseous form. Of course, oxygen is present in other form, the inorganic form, but in oxygen gas, there was no oxygen gas available. Only gases available, probably we estimated that nitrogen and methane type of, and the water vapors, okay, those. And who made this oxygen? There was no oxygen. And how did this oxygen how was this oxygen generated all of a sudden? It's not all of a sudden. During huge, long period of time, like longer than 1 billion years, oxygen was slowly accumulated in the air. Okay. Who did that? That's the point. It's a biologically made oxygen. So out of this prokaryote, only some prokaryotes somehow evolved to learn how to do this photosynthesis. So now you are looking at the first ever form of autotroph evolved out of all 100% heterotrophs. So in the beginning, they were all heterotrophs, but then some of them were able to evolve into autotroph. And this oxygen is simple byproducts of their metabolism. Oxygen is trash for them. And then eliminated those trash out of their body. Now, in the air has more and more and more keep building the oxygen. So it would be really nice if someone could find a way to utilize the oxygen, newly formed oxygen. That's us. Yeah. That story will be continued next week because it's such a long story involving the oxygen. So today I will stop over here. I'm not letting you go, of course not. Because I don't know actually how to handle this. Your questions, there are so many important and interesting questions every lecture I get. But there are only so much time and so many questions that I can handle during the lecture. So probably I will have to come up with some other more creative way of dealing most of your questions. But anyway, so at least taking a very small portion of the lecture time, I'm going to go over some little bit of those. Okay, this one, the question state that, so let's say if you found a new species, then do you have to, every time you found new species, do you have to confirm that through this, the interbreeding, the ability of interbreeding? So if this new species maintain its independency of interbreeding, then now you'll finally declare, oh, this must be a new species. Do you have to go through all those every time? Yeah, maybe that's probably the best way of determining for sure. But let's say this. Wolfer. What kind of wolf is this? This particular wolf is called red wolf. The standard wolf that we know of a gray wolf. In California, and probably some 20-something years ago, I guess, more or even 30-something years ago in California, someone found this gray wolf. I mean, the red wolf. Even there are the brands, the beer brands, having brand name of a red wolf. Okay. So there's been debate going on. So this must be a new species. Okay, red wolf is a new species. We should conserve this new species. This could be soon become the endangered species. So they were so panicked. How do we protect this new species we found, the red wolf? Okay. And then someone came up. Let's say not so fast. Let's try to do this DNA sequencing of these guys. Examined DNA sequence of the red wolf. It turns out that this is gray wolf, regular standard gray wolf. This is what? You don't know anything about this doggy animals? This is coyote. So it turns out that the red wolf was the hybrid, accidental hybrid in nature, between coyote and gray wolf. And once you just found out that, then they just, we don't need to give any extra protection for this red wolf. They will appear and reappear if these two guys have willing to mate again. So this is not really truly new species, so let's not worry about that. So that was the, instead of jumping onto quick conclusion of, hey, this was a new species, so we should do something about establish and protect. But then when they investigate further, it was simply an accidental hybrid. So 2 points. Accidental, this kind of a hybrid between two different species can happen. It's just a biochemical and biophysical barrier exists. There are some specific reasons why these two cannot mate and produce a viable offspring, but sometimes they can somehow overcome those barriers and then hybrid will occur. But naturally, it would be extremely rare occasions. The second thing is, Kayari. The second thing is the DNA sequence. The thing is, how can we determine whether these two are just like someone belong to a single species or not? A lot of times you can kind of estimate roughly or pretty much accurately, you can estimate through DNA sequencing by comparing DNA sequence order between two individuals. Okay, you can somehow give some verdict, pretty accurate verdict. That we are going to handle the logic principle based on how can you use the DNA sequence in determining whether these should be belong to the same species or not, or how can you determine relative relatedness among different species through evolution by simply comparing DNA sequence. There is actually pretty good rationale exists. I'm going to go over later, of course, why DNA sequence can do that. But anyway, this one, artificially, this is also, if there is some kind of a manageable barrier exists, then you can try some artificial interbreeding. But if there is really a tough barrier exists between two different species, there's no way you can do, even if you try artificially. So, yeah. That's a kind of a similar question about, it states that, okay, interbreeding is a criteria for distinguishing two different species, but at the level of genes, how do you determine one from, how do you distinguish one from the other in two different species? Same logic. You compare the DNA sequence, and there is a certain rate. Okay, yeah, this DNA sequence is too different, so it shouldn't belong to the members of the same species. Or DNA sequence comparison gave you some very primitive similarity, so these should be regarded as for same members of the same species. There are certain rules that you can determine. So that's how practically use gene information in the form of DNA sequence in deciding genes or not. Hybrid can reproduce less. Excuse me. Let me jump this whole thing because this is the first actually, the question that I get from the first lecture. So let me directly, for now, skip. I'm not going to simply ignore all of them, but simply because we have only about 10 minutes left. So let me make a big jump into the yesterday's questions. The question asking, this particular question was asking the case of a dog and wolf. Okay. can they be regarded as the same members of the same species or not? They are actually. Dogs and wolves are the members of the same species. Even the scientific name of dog, the wolf is, and probably one N, Canis lupus. The scientific name, like Homo sapiens, meaning human. Canis lupus means dogs. Then what about the scientific name of wolf? Canis lupus. Same scientific name. Although, if you really want to make distinction of dogs only, then you have to put another tail, Canis lupus familiaris family to us. That's dogs. So wolf doesn't have this tail, but usually scientific name system, you have only two things. Okay? Canis, Homo sapiens like. So Canis lupus are the specific scientific name calling for both dogs and wolf. Same thing. Homo sapiens, and those like our relatives who long extinct, the Neanderthals, they were actually able to mate and produce offspring. So by the such rule of interbreeding in distinguished species, Neanderthals and us should have been considered the same individual species. But conventionally, traditionally, without knowing them, before we didn't know that actually Homo sapiens, us, and Neanderthals were able to interbreed. Didn't know. It only just very recently, after DNA sequence comparison between human and those Neanderthals, then it turns out, oh, shoot, we had some mixture of these genes. That means we interbred between. So then it turns out, so then now what do we have to call about? So strictly speaking, of course, by that rule, Neanderthals and us, our ancestors, should have been the same members of the same species. Although traditionally, up until now, they were regarded as a different species. Yes. Yeah, this is really mind-boggling questions. And also another question, similar question is, these days, like humans are not really strictly hard enough selected. What is the selection? Natural selection is actually, what kind of natural selection is coming on us? So unlike other organisms, humans are so like free of natural selection. So what are we going to do? Are we going to evolve into, do we have any chance to evolve into something new under this kind of almost non-existent natural selection pressure? Is another question. Or if you think different ways, what kind of new forms of a human could we ever evolve into? It's another type of similar questions. But now, yeah, actually, unlike other animals or plants or in the past, we do have relatively very less selection pressure coming from nature. Because humans themselves can even control nature. But still, if you think about it, now there is another kind of selection pressure is actually acting on you, regardless, like whether you are aware of it or not. Economic inequality. If you really keep thinking about it, those who have an easier access to all those resources, like economic power, wealth, and those who cannot, rapidly there will be too big of a gap will be existed, and then probably like 100 years from now or 1,000 years from now, there's a chance that because of that accessibility of, because think about it, AI, those who economically afford should be able to freely have an access to and then utilize them to their advantage. Those who cannot because of the lack of money, then probably will behind, okay, in their reform. That's one just a simple example. There are lots of other such risky areas within which you can have such a big gap. Then eventually those will affect number of offspring you can make. And this whole game of, the name of the game is revolution, is how many can you generate. Yeah. Reproductive success is still the goal of the game. So it will be able to affect the different reproductive success between those who and have nots. That's another type of serious possibility. Okay, there's no point of, I'd like to actually go. By the way, I had just 30 minutes ago, I had this. Yeah, what did you do about it? So, was I going to be, no, never mind. Okay, I like, by the way, I like this cartoon. It's very cute and creative. And there was another cartoon I liked. This explained the natural selection so well. And another thing is actually, oh, sorry. Oh, I didn't include, sorry. Okay, I will just pick it up next week, the rest of it. Okay, have a nice weekend and see you on next week. Bye.